Okay, in this video, we're going to go over platelets and blood clotting, so how platelets work to stop bleeding. All right, so the first topic is we're going to define the word hemostasis. And so hemostasis just refers to the process of stopping bleeding. All right, so that's the function of these platelets, is to stop bleeding or to cause hemostasis. Okay, and so the platelets are derived from, if you remember, we've talked about it a couple times now, these hematopoietic stem cells. All right, so that's our first cell, and then remember platelets. We had our two branches, the lymphoid stem cells and the myeloid stem cells, and platelets come from that myeloid stem cell. All right, and then that myeloid stem cell is going to become a giant cell that is called a megakaryocyte. All right, megakaryocyte. Okay, and this is the type of cell that makes platelets. And so it makes platelets by producing all the chemicals that are going to be found in platelets and pinching those off in these little vesicles. All right, so these will just be little balls of plasma membrane that are full of different chemicals. And we'll talk about some of those components in just a minute. So these would be the final platelets that were made. Okay, so again, platelets are not actual cells. They're just little cell fragments, and the cell that they're fragments of is a megakaryocyte. All right, so megakaryocytes make platelets. Okay, so then two words pertaining to the abundance of platelets. The first being thrombocytosis. So this would mean that you have a lot of platelets. All right, so many platelets. Okay, and so somebody with thrombocytosis, you might be concerned about clots forming when, there's no necessary reason for clots to be forming. All right, and then there's thrombocytopenia. Okay, so this is too few platelets. And so someone with too few platelets, you would be concerned about bleeding or them not being able to stop bleeding if they have like a wound or something. All right, so excessive bleeding. Okay, and so then in platelets, we said they're kind of like cell membranes full of different chemicals. And so there's three chemicals that you guys should know, components found in platelets. So the first two, ADP and thromboxane. All right, actually I'm going to highlight those. Okay, so ADP, thromboxane A. Those are the first two. So ADP actually is like adenosine diphosphate. Okay, so it's kind of what's left of ATP, except it has a function totally separate from ATP. Okay, and thromboxane A, both of these have a very similar function in that they promote other platelets from sticking or to stick. All right, so promote platelet sticking. Okay, and so this is important because when those platelets are going to stick to some sort of wound or rupture in a blood vessel, the platelets are going to stick to that open area in the blood vessel, and they need to recruit more platelets to come and stick to that area. So you get this kind of buildup, this almost like wall or barricade of platelets. Okay, and so ADP and thromboxane A are going to promote those platelets coming in and sticking along with the one that's releasing these chemicals. All right, and then serotonin is also found in platelets. This is the same serotonin, that's a neurotransmitter, except here its function is vasoconstriction. All right, so vasoconstriction. And that's important because obviously with bleeding, there's a wound in a blood vessel, and so blood is leaking out of that blood vessel. So if you can constrict that blood vessel, you can kind of reduce temporarily how much blood is being lost. Okay, so immediately platelets are going to release serotonin to help kind of constrict that blood vessel and reduce the amount of blood being lost. Okay, so now let's talk about the process of hemostasis or blood clotting. All right, so the players involved are going to be clotting factors. All right, so clotting factors, these are going to be found in the plasma. All right, so they're just dissolved, floating in the plasma, clotting factors. We'll talk about some of them in a little bit and what they do. And then there'll be the substances from those platelets that they're going to be releasing. So that ADP, the thromboxane A, and the serotonin we just talked about. Okay, and then also there are obviously going to be some injured cells. Okay, and so those injured cells of the blood vessels that are torn or broken are going to release chemicals that normally would be in their cytoplasm, and those chemicals actually will kind of trigger this process, this hemostasis, or at least they play a part in it. All right, so before we talk about the steps, one additional thing I want to touch on is endothelium. Okay, so the endothelium is the tissue that lines the inside of the blood vessel. All right, and so remember that epithelial tissue on the inside of the blood vessel is simple squamous. Okay, and so the job of this endothelium is to provide a extremely smooth surface on the inside of the blood vessel. All right, and so it is gonna, so essentially it's kind of like a non-stick coating on the inside of the blood vessel. So smooth surface. All right, and so smooth surface inside the blood vessel. Okay, whoops, little pencil glitch there. Okay, all right, and so the reason for this is because platelets will stick wherever there is some kind of uneven surface, or maybe there's like some collagen exposed, there's a bump or a tear, anything like that, anything that is not nice and perfectly smooth, platelets will start to stick there. Okay, so the endothelium's job is to make this inside of the blood vessel very smooth so platelets don't stick. They just move on through with the plasma. All right, and so in order to do that, the endothelium secretes two chemicals. All right, so prostacyclin, which is a type of prostaglandin, and nitrous oxide, which is a gas. Okay, and so both of these together gives the endothelium, the inside of the blood vessel, a non-stick coating. Okay, so they just kind of coat the inside of that blood vessel and keep platelets from sticking. All right, give blood vessels a non-stick coat. Okay, and so when that kind of coating, that barrier of prostacyclin and nitrous oxide is disrupted, then we'll start to have platelets sticking. Okay, so let's talk about our stages. So the first stage is vasospasm. Okay, and with vasospasm, the blood vessels are going to constrict. So the first thing that's going to happen is that the blood vessel, whatever blood vessel is damaged, is going to constrict. Okay, and so we can see that here. All right, so we have the damage in our blood vessel, and so immediately that blood vessel, the smooth muscle, is going to start to squeeze and kind of shrink that diameter, all right, of the blood vessel. And so this will decrease blood flow. And that will last for about 20 to 30 minutes. Okay. All right, so this is the initial step to kind of, mitigate the possible disaster of blood leaking out of the blood vessel. And so it happens in response to damage to the endothelium, which we were just talking about. So if that endothelium is damaged, then that will trigger vasospasm, the blood vessel to constrict. If the smooth muscle in the wall of the blood vessel is damaged, right, so that would be a very deep wound, that would cause it to constrict as well. And the triggering of pain receptors in the wall of that blood vessel would also cause vasospasm, as well as, pain receptors in the surrounding tissue. All right, and so this vasospasm obviously would be most effective at stopping blood in very small blood vessels, but in any of the blood vessels, it's going to at least slow that loss of blood. Okay, so after vasospasm, we start to have platelets finding that open area and sticking to it. All right, and so what's going to happen is they're going to form a platelet plug. Okay, so for this, the platelets will stick to the uneven surface. All right, and so there would be exposed collagen in that area, those damaged cells, that endothelium would be broken, and that layer of prostaglandin or prostacyclin and nitrous oxide would be disrupted. Okay, and so that would trigger platelets to stick. All right, and so platelets sticking, that's called adhesions. Adhesion. All right, so platelet sticks, that's adhesion. Okay, so first they adhere, and then they're gonna release. Okay, so once they adhere, they become activated, and then they're gonna release those contents we talked about. So that ADP, that thromboxane A, right, and that serotonin. Okay, so all those things, remember serotonin is gonna, what is going on here? All right, that serotonin is going to cause further vasoconstriction, and the thromboxane A and ADP are going to attract other platelets, okay? And so more and more platelets will come in response to those signals, and that will cause aggregation, okay? And so aggregation would just be the further accumulation of those platelets, and so that is going to form a temporary, because we're going to have a permanent plug in a little bit, plug. Okay, so our platelet plug is kind of like a temporary band-aid. If you have like a wound, right, that's kind of like a band-aid you would put on it, and then maybe you need stitches, so then you go to the ER and get stitches and get a more permanent solution. Okay, so this is our temporary plug. All right, and then the third stage is coagulation. Okay, and so this one's a little more involved. So with coagulation, this is where we're going to form that clot. And so the clot is, so we're going to form the blood clot. That clot is going to be the permanent plug. Okay, so that's the goal here, is to form that permanent clot. Okay, so our permanent plug. And so it is made up of a mesh, kind of interwoven, insoluble fibers, right? So that means that they're not soluble in water, all right? So they're insoluble fibers. And so we're going to just make this big mesh, all right? You can kind of see it here, right? It's this woven kind of yellow mesh of insoluble fibers that are going to span that whole broken area and plug it up. Okay, so we'll just put that here, all right? So it's a mesh of insoluble fibers. And so those fibers that we are going to end up with, they're called fibrin. Okay, and so you may remember when we learned about the composition of blood, one of the plasma proteins that is dissolved in the blood is fibrinogen. Okay, and so during this process, very last thing we're going to do is activate that fibrinogen to fibrin so it can form this platelet or this clot. Okay, so let's talk about how that happens. And so the way that happens is through a cascade reaction. All right, so we're going to have a few of these in A and P2. And so a cascade reaction is where you initially, whatever the signal is, like this damage, right, to the blood vessel, activates something. All right, usually it's going to be an enzyme. So in this case, it is an enzyme. It ends in ASE. That means it's an enzyme. All right, so, and then that enzyme will activate a next step. The product of that step will activate the next step, and so on until you reach whatever your final goal is. Okay? All right, so our first step is to activate and form that prothrombinase. All right, and so it's kind of complicated. All right, and there's a lot of steps. There's like 2 pathways. There's an extrinsic and an intrinsic pathway. I'm not going to go into all that detail about that. If you want to read about it, you certainly can in your textbook, but I'm not going to test you on it. All right, just know, so the extrinsic pathway would just mean it's coming from outside of the blood vessel. So signals from tissues damaged outside of the blood vessel are going to contribute to activation of this prothrombinase. All right, so that's one way. And then intrinsic would be from inside of the blood vessel. So those platelets that are releasing chemicals, the collagen that's exposed, the endothelium that's damaged, that is going to activate the prothrombinase. So it can, and they actually work together. All right, but so they're going to be activating the prothrombinase. All right, and so many of these signals are clotting factors. All right, and those clotting factors, as we talked about with plasma, are just floating around in the plasma. Okay, and one thing to note that I do want you guys to be aware of, we'll talk about it towards the end, is that the clotting factors in the plasma are, there are, was it 13 of them total in the plasma? 4 of those are derived from vitamin K. Okay, and they're made by the liver. Okay, so it's important to have vitamin K in your diet in order to be able to clot properly. Okay, so we have prothrombinase has been activated by any number of these signals that are signaling damage to surrounding tissues or the blood vessel. All right, so now that prothrombinase is going to go and activate the next step. Okay, so this prothrombinase will activate and cause prothrombin to be converted into thrombin, which is its activated form. All right, and so then that thrombin is now going to go and activate fibrinogen to fibrin. All right, we just talked about fibrin is that insoluble fiber. All right, once it's activated, it's insoluble fiber, and we'll add that here. So it's an insoluble fiber, all right, meaning it doesn't dissolve in water. And what it does is it binds with a bunch of other fibrin molecules, and they make the plasma in that area very gel-like. Okay, so it kind of changes the consistency in that area, which is going to kind of block, right, and kind of form that permanent clot in the blood vessel. Okay, and so that fibrin all polymerizing, and then they will interweave. Okay, so it's going to weave into the platelet plug. So all those fibrin that have been activated. All right, so we're going to weave into the platelet plug. Okay, and so that's what we have here. All right, so we had our platelet plug. So our platelets, remember, we're releasing all those chemicals that attracted more platelets, and they all stick to form our platelet plug. Okay, and so then the last step of coagulation, we had our fibrin that had become activated, and it will weave into that platelet plug, kind of reinforcing it and forming that permanent clot. Okay, good. So we have our permanent clot, all right? So then that will give the tissue time to heal, all right? And so one thing that happens with that permanent clot is it's called clot retraction or syneresis, okay? And there are actually, and you learned about actin and myosin and ANP1 with muscle contraction, there are actually actin and myosin proteins in this clot, and they're going to start kind of, grabbing those fibrin fibers. They're like long threads, and they're going to start pulling them together. And so what this does is it pulls the damaged edges together, right? So of that blood vessel that's damaged, all right, so damaged edges together, okay? And so that's going to help to bring those two pieces closer to each other, making it easier to kind of repair that wound. Okay, so that's clot retraction. All right. So then once the wound has completely healed, the process of getting rid of that clot when it's no longer needed is called fibrinolysis. Right? So we have fibrin, which is making up that clot, and then lysis means you're going to be breaking something. So we're going to be breaking up that fibrin. All right, and so to do this, we are going to need plasminogen, which is a component that is incorporated into the clot when it's formed, okay? All right, so this is added to the clot when formed, okay? So it's kind of adding a way to get rid of that clot when you're making it, okay? So you're going to add plasminogen to the clot when it's formed, and so then once that tissue is fully repaired, this plasminogen is going to activate to plasmin, and that plasmin is going to digest fibrin, all right, and break it down, okay? And so when it does that, that's going to dissolve that clot, and that clot will be gone, all right? Okay, so that is hemostasis, and so we just have a couple more terminology topics to talk about, and then some clinical implications of clotting, and we'll be done with the blood chapter. All right, so a couple terms you've probably heard of, a couple of these. All right, so first of all, a thrombus. This is going to be a clot, and it's specifically referring to a clot that is attached to a vessel wall. Okay, so this would be an intact kind of clot on a vessel wall. Okay, and so then if for some reason that clot detaches and is now floating in the bloodstream, that is referred to as an embolus, okay? A clot, so this is a clot detached from the vessel wall. Okay, and so it's called an embolus when it is floating in the blood. Okay, so this is floating in the blood, and it can, as I'm sure you've heard, get lodged in very small blood vessels, and so then at that point it's called an embolism. All right, so when it's stuck, and so usually when it's stuck, it'll be stuck in a very small blood vessel, right? That embolus is too large to move through. Okay, and so you can have pulmonary embolisms, and you can have arterial embolisms, okay? And so a pulmonary embolism, and this will make a little more sense when we go through the circulation of the heart, all right? So just keep this in mind when we do this, but a pulmonary embolism is going to form in veins, okay? So it's going to form in a vein because venous blood is being returned to the heart, all right? So venous blood blood in veins doesn't have oxygen, so it's going to be returned to the heart, so then it can be sent to the lungs so it can get oxygen. So it's not going to go through any capillary beds. If there is an embolus that forms in a vein, breaks off, it's going to float. It's not going to encounter a very small blood vessel until it actually reaches the lungs, where that blood would pick up oxygen. Okay, and so then that embolus would lodge and become an embolism in the lungs. All right, that's why it's called a pulmonary embolism. Okay, and then you can have arterial embolisms. So this would be an embolism in a smaller capillary out in the tissues. All right, and so this would start in systemic blood. All right, so forms in like an artery. Systemic, well, actually, let's say, yeah, so it forms in an artery. Okay, and then it would get lodged in a very small artery or capillary. All right, so this would be out in the tissues. So you could have an arterial embolism maybe down like in your leg or your foot or something like that. All right, so small artery / capillary. Okay, good. And so some causes for embolisms. So one major cause is when there's a decrease in blood flow. So blood just isn't moving very well. Okay, so there's a decrease in blood flow, and sometimes blood maybe will start pooling especially in like the extremities. Okay, and so you worry about this with like bedridden patients, or if you're going on a really long flight. That's why they say you should get up and walk around, and that is because your veins, one of the ways that your veins get blood back to the heart, right? If you have think of a vein down in your calf, it's fighting gravity to get blood back to your heart, and it does that by the skeletal muscle contracting and helping to squeeze that blood back to the heart. And so that is referred to as skeletal muscle milking. Okay, so if there's a decrease in skeletal muscle milking, all right, all of these things are kind of associated. So if we have a decrease in skeletal muscle milking, or somebody is sedentary for a while, that would mean that they have a decrease in skeletal muscle milking. The blood will pool from that decreased blood flow. And when blood pools and is not moving very well in circulation, it will start to clot. Okay, if it sits too long, still for too long, it will start to clot. All right, and so then those clots will form an embolus that could become an embolism if it gets lodged somewhere. All right, so our last topic are some clinical implications of these things we've been talking about. Okay, and so some medications you've probably heard of are heparin and warfarin, and so I'm just going to mention them in a sense, so now you'll actually understand how these work. Okay, so heparin specifically is going to block thrombin. All right, so these are blood thinners. Okay, so it's going to block thrombin. And so if you remember, thrombin is what activates fibrinogen to fibrin, and so fibrin is never activated. Okay, so this is a blood thinner, meaning fibrin is not available to form that clot. Okay, whoops, I meant to say it's a blood thinner. I got distracted by my own words. There we go. All right, so it's a blood thinner. Warfarin, also a blood thinner, however, it acts a different way. So warfarin is a vitamin K antagonist or blocker, right? So it's kind of a vitamin K blocker, right? So antagonist. Okay. And so what it does by blocking, remember vitamin K is really important for development of and it's what these clotting factors are derived from a certain number of clotting factors, right? So we have a decrease in clotting factors being produced, and that is going to lead to kind of a blood thinning effect, right, if you don't have those clotting factors. Okay. And then we have hemophilia, all right? So hemophilia is a genetic condition, all right? And it's a genetic condition where someone is actually missing the proper genes for certain clotting factors, all right? So that person is missing clotting factors. Okay, and that would be, again, a problem with their DNA, and so they would not be able to control if they got a wound. It really wouldn't clot and heal up that well. Okay, so that could be, a concern, especially internal damage that you can't see the actual wound bleeding. That could be very dangerous. All right, so then our last little topic here is disseminated intraventricular coagulation, or DIC. Okay, and so DIC is very rare, but it's interesting to talk about. So it is systemic activation, all right? Systemic. And so if there's one thing I want you to take from class A&P2, you're going to understand what systemic means when you see it. So it's systemic activation of coagulation, right? So clotting. Okay, and so systemic means that is happening all over the body, right? It's not localized to a specific area. Systemic means it's happening everywhere. So everywhere, all at once, coagulation starts to happen. All right, so obviously that is a huge concern, because then you would have all of these embolus forming, and they'll go and form all of these little emboli. All right, and that would be very, very dangerous. Okay, so some causes of this would be there are some systemic infections and sepsis that can trigger this. All right, so trigger systemic coagulation. Very large amounts of trauma to the body can trigger this DIC, as well as certain cancers have been known to trigger DIC. Okay, and so then it would lead to what's called a microvascular thrombi, these tiny little thrombi forming in the very, very tiny blood vessels. Okay, good. All right, so that is the end of the blood chapter. And so then our next chapter will be the heart, and then that will be the last chapter for your first exam.